This sets up a new section, S1P3, Section 1, Part 3, which are system examples in homeostasis. If you remember back in one of the earlier lectures, we discussed the human organ systems. We have 10 that we're supposed to remember for this course. 5 of them I mentioned were going to be studied in a fairly detailed overview of them, while the other ones are much less detailed. And so we're going to get most of those out of the way with this next lecture or 2. So this is a material for lecture exam 1. Let's go ahead and get started. So remember that this course builds upon previous knowledge, both in this class and anatomy and chemistry as well. So we're going to call back on some of that knowledge maybe in this lecture today. So let's start with the digestive tract. That's going to be obviously the digestive system. And most of the time when we talk about the newer systems and introduce them, we're going to start with the function. So the primary function of the GI or gastrointestinal tract is to move nutrients, water, and electrolytes from the external to the internal environment. So we have to have some way to get them into the body. And in order to do that, we require the coordination of 4 basic processes to maintain digestive homeostasis. And our body must do that to work together for normal function. Okay? So the 4 processes we're going to worry about, and you see different books name them this way. This is how you're responsible for it on our exam, would be digestion, absorption, motility, and secretion. Digestion sometimes is a confusing element because in some ways, from especially a lay point of view, digestion refers to the whole process we're going to talk about. But scientifically, digestion really refers to breaking particles into small enough pieces in order to absorb them. We can break those down 2 ways, mechanically and chemically, and we'll talk more about those in a minute. Absorption means to take them from the external to the internal environment to somehow get them into your body. Motility refers to the tract itself. We have to move food that we ingest in our mouth, move it through our digestive system, which is somewhere between 6 and 8 meters long in most humans. And the things we don't absorb, right, go out at waste. And then the last one is the idea of secretion. And secretion is when we release certain chemicals to try to help the process along. So we're going to look at those in some detail here. So digestion, again, is breaking food particles in small enough units so that we can absorb them. It's a physical process as well as a chemical process. The physical process is sometimes called mechanical digestion. Often involves the teeth, right, through the process of chewing. Scientifically, that's known as mastication. And the idea is to break those particles that you eat into small enough units so you can swallow them and eventually start the digestive process through this mechanical means. You can also have some digestion mechanically in the stomach. Here's our stomach right here. Stomach's usually considered a J-shaped organ with 3 muscular layers. And the chemical digestion also happens here and also happens in the mouth, but mechanical digestion happens here as well. And that's through a process called churning. Now, churning to a large extent is to move the food and kind of turn it over and expose the food particles to other portions so that you just don't digest the outside, but you constantly turn over the contents. The process of doing this does crunch the food a little bit and does a little bit of mechanical digestion. But most of the mechanical digestion is through chewing. Also, when the food stuff, which is known as a bolus, gets into your colon, you also have something called segmentation, which is a little bit of mixing, exposing the surfaces to other things, and also a little mechanical digestion as well. So those are the physical processes. The chemical processes uses secretions, primarily exocrine secretions, to break down food. Chemical digestion happens, actually starts in the mouth and with something called salivary amylase. Salivary amylase, well, every time you see the word amyl, it refers to carbohydrate typically. And ACE often tells us it's a protein. Not everything that ends in ACE is a protein, like suitcase, if you're going on a trip, wouldn't be. But biologically, if you see a word there, you ought to suspect that it's a protein. And so salivary tells us it comes from the mouth, right? That makes sense, saliva. And so salivary amylase is a chemical that breaks down carbohydrates in the mouth. Now, when we eat carbohydrates, remember from the biomolecules, we can eat carbohydrates in a couple different forms. They can be polysaccharides, like starch, as an example. It could be a disaccharide, so a simple sugar, but in 2 units. The disaccharides would be like sucrose, as an example, the table sugar you would put in a coffee or something. And then it could also be a monosaccharide, like glucose. So as we eat these larger carbohydrate molecules, we start to break them down chemically with these salivary amylases. Also, we have mucus that really doesn't start to break down the food so much, but to help lubricate the food and provide a little bit of buffering so we don't have these huge changes in pH yet from that. So chemical digestion also, further occurs in the stomach through a number of different things. We start to release other chemicals in the stomach. One I'm sure you're all familiar with, hydrochloric acid, HCl, comes from the stomach. The acid tends to break down a number of different types of molecules. We also have what are called proteases. So proteo is referring to proteins, and aces, again, enzymes. So proteases begin to break down the proteins into their amino acid constituents, so into smaller, basically peptide sequences. The main proteases you see in the stomach would be things like pepsin, and so pepsin's a good example of that as it starts to break down proteins. And then chemical digestion also occurs in the small intestine when we start to have our lipases, so lipid, right? So lipases break down lipids into their subunits, and that helps with the bile salts and emulsification and a few other things like that. But, the chemical digestion occurs for the most part throughout most of the digestive system, especially in the earlier parts. So carbohydrate digestion begins in the mouth with salivary amylase. Protein digestion begins in the gut with proteases, and a great example of that would be a pepsin. And then the lipid digestion, right, begins in the small intestine with our lipases. Right. So that's digestion. Absorption is the next one we're going to talk about. And absorption, again, is transferring substances from the external environment to the internal environment. And when you think about it, the digestive system is really just a tube that runs from one end of your body to the other. And technically, it's sort of the outside of your body, right? So even though it's inside, it's communicating with the outside world. So in order to get it into our body, we have to take it from the lumen, from the place where the food goes to, and bring it into the body. All right, so we do that with a variety of processes, and here's just some examples that I'll mention. So first of all, water-soluble materials. That would be things that are polar. Polar things would be our nucleotides and nucleic acids, our amino acids and proteins, our carbohydrates and our monosaccharides. Our minerals typically come in ionic form, so those would be water soluble. Many of your vitamins are water soluble, not all of them, but many of them. So our water soluble materials, since they're polar and water is polar, they will dissolve in blood because blood is more than half water. The water portion of blood is called plasma, and most of plasma is water, and water plasma makes up at least about half your blood, generally speaking. And so what happens there is we absorb things from the lumen of the gut, right? Especially in the small intestine, beginning of it, the duodenum, or duodenum, however you want to say it, and directly through the walls of the epithelial tissue here and into the capillaries. So water-soluble materials are absorbed directly into the capillaries. We can't do that with fat, though, because fat and water don't mix. And what happens is the fat would precipitate out because it's not soluble. And you can't have fat precipitate into your blood vessels, otherwise it would cause problems with flow and things like that. So fat-soluble materials like lipids and your fat-soluble vitamins, A, K, E, D, would have to be absorbed through a different route. And what we do there is we absorb through what's called the lacteal. So the lacteal right here points to this little green structure right here. Okay, and the fat-soluble materials go into the lacteal, and then the lacteal is actually part of your lymphatic system. And those go to the liver processes it, puts a protein on it so that it can be soluble in blood, and then sends it into the blood. So as an example, you would absorb cholesterol this way. So it's soluble in the blood and doesn't precipitate out. So the release of bile salts along with some of those lipases helps to break these particles up into small enough units where they can fit into the lacteal. And then the amylases and proteases help break down those. And there's also nucleases for the nucleotide, to get nucleotides from the nucleic acids. Those are all broken down into small units so they can be absorbed here. One of the things, which isn't listed here, but it used to be thought that we had to completely break down all the materials into their basic unit in order to absorb them. So proteins had to be broken down into individual amino acids. Carbohydrates had to be broken down into monosaccharides. Lipids had to be broken down into fatty acids and things like that. But we're finding that we can absorb, in certain cases, some small molecules that we can just take into the body. There's some evidence that says that might be the reason why people have food allergies, that you absorb these small molecules into the body, and now because they're not single units, but they're multi-units, your immune system reads those, sees them as foreign, and decides to attack them. And those have similar structures to, say, peanut butter or, like shellfish, things like that. So who knows if that's really true, but it seems like, bottom line is we don't have to absorb things as single units. Vitamin B12 is an interesting component of this, and not everything gets absorbed this way, and sometimes it's just absorbed by what they call mass action. So as an example, your amino acids are absorbed somewhat randomly. Because they're polar molecules, they can't pass directly through the cell into the capillary, so they have to be transported. And the amino acid transporters just pick up an amino acid. So your amino acids, to a large extent, get absorbed at the rate at which you're eating them. So if you ate a ton of lysine for some reason, then your body would absorb a ton of lysine because it would outcompete the other amino acids for binding sites and be able to be taken into the body. But some of the materials that we absorb are absorbed a little differently, and here's a couple examples. One is vitamin B12, as I mentioned. It's scientifically sometimes known as cobalamin, and you have intestinal transporters for vitamin B12 in the ileum, right? So, going back to anatomy, right? You have the mouth, the pharynx, the throat, the esophagus, right, for the digestive system. That leads to the stomach. That leads to the small intestine. The small intestine is made up of 3 sections: the duodenum, the jejunum, and the ileum. And so in the ileum, we absorb cobalamin. And what's interesting about that is, in order to absorb vitamin B12, we need a cofactor, another chemical present, to allow us to do that. And that chemical is called intrinsic factor. Intrinsic factor is made by the same cells in the stomach, that the parietal cells, that make the acid, the stomach acid, the HCl. And so intrinsic factor has to be there in order to absorb vitamin B12. What's interesting is some people don't make intrinsic factor as well, and they start to become vitamin B12 deficient. You could have damage, or it often occurs with age, and especially prevalent in people who drink really, really hot liquids. So as an example, if you visited Starbucks every day and got an extra hot latte, then you would increase your risk for this happening. The reason why this is important is vitamin B12 is essential to make red blood cells. So people who don't absorb vitamin B12 cannot make sufficient blood cells, and typically they become anemic. Right? Anemic is a lack of ability to carry oxygen, and one of the primary, not the only, one of the primary causes is a decrease in the red blood cell number. So not being able to absorb vitamin B12 could affect that in a negative way. If you found somebody that had that issue, what they do is they just bypass the absorption of that, and you just give people a shot of vitamin B12, and that gets everything back to normal. There are a few other substances that are absorbed in the gut that are regulated, and calcium is a good one there. Calcium is absorbed through what are called calcium channels, little protein channels within the membrane, and calcium absorbed that way. And calcium is regulated, its absorption, by vitamin D3. And so vitamin D3 is essential for the absorption of calcium. So if you were to see a calcium supplement or look at milk or most milk products, it'll say that it's fortified with vitamin D. And that's because it will help you absorb the calcium in that tablet or product that you're consuming. So calcium needs a substance that regulates it, and that's vitamin D. Iron is also interesting, and it's regulated in a little bit different way. There's actually 2 forms of iron that we can absorb. What is called heme iron, which comes from things like meat products, and then ionized iron that comes from some plants. Both eventually need to get into the cell by a protein called ferroportin. Okay, so that's what this little red circle is supposed to represent. Here is the stores, and so it's hard to see, but that's this little green thing here, would be the actual protein transporter. And ferro, right, refers to iron. Like, so, I'll give you a second to think about what's the chemical symbol for iron. Right? I think most of you probably realize it's Fe, right, for ferrous. So ferroportin is actually not a bad name if you break it down. Ferro refers to iron, and portin is what it sounds like, transporter. So ferroportin is this transporter that allows you to absorb iron from your gut. So when you have need iron, then what you do is you don't make a whole lot of this chemical called hepcidin. Hepcidin is released from the liver, and what it does is it destroys the ferroportins. So it basically inactivates these transporters. So when you have low hepcidin, then you have these functional ferroportins which allow us to absorb iron and put it into the blood. Right? I think most of you probably realize that iron is an important constituent of red blood cells, and in fact the most common form of anemia is iron deficiency anemia, where you don't have enough iron for various reasons. When we get to the blood section, we'll talk a little bit about that, so we won't worry about it now. Hepcidin helps to control the activity of the ferroportin. When you have a high level of hepcidin, right, when you have a sufficient amount of iron typically, then the hepcidin basically inactivates, turns off the ferroportin, and you don't transport iron and you don't absorb nearly as much as you would normally. So, it's another case of another chemical helping to regulate the absorption of something into the body. So there's just a couple examples and kind of show you some of the differences. Some things are unregulated, some things are regulated with another chemical like D3 and vitamin and calcium, or hepcidin and iron, and or intrinsic factor allowing you to absorb vitamin B12. So that's absorption, taking things into the body, typically from the gut, right? The third one is motility. Sometimes that's called propulsion, right? So to propel something through the digestive system. I probably should change the order here, right? In terms of motility, right? One of the first portions of motility we'd see in the digestive system would be swallowing. Swallowing is known as deglutitation. Okay, so that's swallowing, where you take that bolus of food and move it into your esophagus. Throughout the digestive system, the food moves through peristalsis. So peristalsis is rhythmic contraction behind the bolus, right? Relaxation in front of it, and that kind of squeezes the bolus down. So here's showing the esophagus leading into the stomach, but it does a similar thing in the intestines as well. So peristalsis is important. And then to get rid of the waste product, right, would be defecation, right, and sending the undigested material, the unabsorbed material. It could be digested, broken down, right, but the unabsorbed material out of the body. And again, we saw some examples of secretion already. We talked about enzymes and things like that, but secretions can come in a whole bunch of different forms. Hormones, we're finding there's a bunch of hormones that help regulate this. Leptin, ghrelin, CCK, cholecystokinin are all examples of hormones. You have to worry about them. You don't have to worry about what they do. I'm just telling you there's hormones. Enzymes, we talked about some of them. The proteases, the lipases, the amylases, right? We mentioned mucus that helps to lubricate and buffer the food. I mentioned bile and producing biosalts and helping to break down fats. Paracrines. Paracrines are chemicals that are released to work on local cells. So paracrine means local or nearby chemical. So paracrines work on other cells. So those are important. And there's a bunch of other things as well. So secretion just helps us to control the whole process for that. Since we talked about calcium, let's talk a little about bone tissue and homeostasis. When we look at the levels of calcium in the body, calcium is a homeostatically regulated variable, meaning we should have normal calcium levels in the blood. We'll talk a little bit about the importance of calcium for muscle contraction. We'll talk a lot about it in lab. We'll talk a little bit about it for skeletal muscle. We'll mention a little bit of it for smooth muscle as well. So calcium and maintaining the normal calcium in the blood is very important. So in order to help control this, we store most of our calcium in our bones. Right? So if calcium levels go up, then the thyroid releases a hormone called calcitonin. So basically it means it maintains the calcium tone. That increases calcium deposition in the bone, right? Because if calcium goes up, we want to bring it back down to normal levels. So since the calcium's in the blood, we need to figure out something to do with it, all right? And so we want to decrease blood calcium because their levels are too high. So there are three things we can do to affect that. Is one, increase the calcium deposition in bones. We can store more calcium in bone, get it out of the blood, store it in bones. We can decrease our calcium uptake in the intestine. So we don't take in as much calcium from what we eat, which means that the calcium in our blood lowers. We can also decrease the calcium reabsorption from the urine. Okay? So normally we pull out calcium out of the urine, and not all of it, but some of it. And so if we quit doing that, there'd be more calcium in the urine, which means you'd pee more calcium, and that would lower the blood. So calcitonin does that, and it brings calcium levels back to normal. If calcium levels went down, we would release a different hormone. This different hormone would come from the parathyroid glands. There's 4 of them typically, kind of surrounding your thyroid gland. And that releases a very nicely named hormone called parathyroid hormone. And so if calcium levels go too low, we release parathyroid hormone, and it's antagonistic to calcitonin. It does opposite things. So what parathyroid hormone does, because calcium's too low, we want calcium, we'll look over here on the right side, levels to increase in the blood. So the way to do that is to take the calcium that's stored in the bone and to release it into the blood. That'll raise it. To increase the calcium uptake in the intestine. So we're going to take in more calcium in the intestines so that we can increase the blood levels. And we're going to increase the reabsorption of calcium in the urine. So what we would have peed out, now we're going to pull out of the urine and save. So calcium is regulated that. Phosphate is similarly regulated. In fact, a lot of our phosphate is stored in the bones as well. But we won't worry about the details for phosphate, but just know that the bone stores a lot of our calcium and a lot of our phosphate. All right, so where's the calcium? I mentioned you stored a lot of your calcium in the bones. Our bone reservoirs are about 99%, so almost all of your calcium is stored in the bones. About 1% is intracellular. We haven't talked about intra and extracellular yet, but intra means inside. So we have about 1% of our calcium stored inside of the cells. And then our blood and our other extracellular fluid compartments, like our interstitial fluid, is about 0.1%. So 0.1% is our calcium that's not in the bones and basically not inside cells. So fluxes in calcium and phosphate are controlled, as we kind of saw when we went over that homeostasis for calcium figure, by 3 places: the small intestine, the bone, and the kidneys. The small intestine, because it dictates how much calcium you have coming into the body. The bone, because it dictates how much calcium is stored. That's where almost all of it is, right? Stored in the bone. And the kidneys, which dictates the reabsorption of calcium, basically how much you pee out in your urine. So by balancing those three things, we can balance calcium in the blood and hopefully in the bones as well. Now, we mentioned these already, so just as a quick review, I'm not going to go through it step by step like we did before, but there are basically three things that help in terms of hormones control calcium. All right, I probably should have these in a different order too. First of all, vitamin D, because vitamin D, remember we talked about the absorption of calcium, right? So vitamin D helps us to absorb calcium. So the more vitamin D you have, the more calcium you're going to absorb theoretically. Calcitonin, right? We saw calcitonin helps to decrease the calcium in the blood back to normal. And then parathyroid hormone, if calcium drops, it helps to increase the calcium back into the blood, right? By affecting what is called osteoclast activity. Those are the specialized white blood cells. They're a type of macrophage called osteoclast that basically degrade the bone and release calcium back into the blood. And they're stimulated, their action, by parathyroid hormone. All right. So phosphate, again, is like 85% stored in your bone, while calcium we saw on a slide ago was 99%. So it's similar to what happens with calcium. And again, we're not going to worry about the details. Let's talk a little about muscle. This should be mostly a review here for muscle. These aren't the greatest pictures. Their resolution's not as good as I would hope, but I sort of like them from a couple different standpoints. I wish they were flip-flopped a little differently, but I like the fact that we have cardiac muscle in between smooth and skeletal muscle. And the reason why I like that is that cardiac muscle in many ways behaves like smooth muscle, and it has some of its own things, but looks like skeletal muscle. So it's sort of this intermediate type of muscle we'll talk about. And so it has many of the attributes of smooth muscle and some of the, at least functionally, and some of the structurally same characteristics as skeletal muscle, and then a few of its very own, like this little purple thing here and here, the intercalated discs. All right, so I'll look at it in kind of detail. So let's start with skeletal muscle. Now, skeletal muscle, you've probably studied in your anatomy class in some detail, but you probably didn't go over these. And the reason being is your anatomy class focused for the most part, I'm guessing, on human anatomy. Most of the college courses go into human anatomy. And so you might have not talked about this first type of muscle. And there's a couple reasons I want to talk about it. One, because, it is represented a little bit in humans, not much at all, but we do have some. So it's probably nice to know about that. The second thing is probably more important from a class component. We're going to talk about this muscle type when we look at the frog lab. And so it's nice to know about it in lecture and be introduced, so it'll be easier to understand when we get to that laboratory component. So skeletal muscle has a couple of different characteristics, but one of them is that it's voluntary, right? It's also reflexive, but it's largely voluntary. So tonic muscle is like skeletal muscle under voluntary control, but it behaves a lot like smooth muscle. Okay? It is very aerobic. It has a ton of mitochondria and a ton of energy storing capacity. And the reason why you probably haven't ever heard of it before is humans don't have much of it. We really have it in just a couple places. One is the orbicularis oculi. Okay? So if you remember that muscle, it's the one that goes around your eye. And some of you guys sleep 14, 16, 18 hours a day. And when you sleep, what happens to your eyes? They stay shut, right? So for things that need prolonged contractions, sometimes in some species, they'll have tonic muscle. So tonic muscle is really designed for these sustained, long, tonic contractions. So we have some tonic muscle in our eye, nabicularis oculi. And not surprisingly, our sphincter muscles within our urinary and digestive system also have some tonic muscle, which allows us to close off a sphincter for extended periods of time, which especially socially is a nice thing to be able to do. Right? Sometimes that's called slow muscle or non-twitch muscle. And it's called that because it takes a long time to develop tension, it takes a long time to relax, and you can't stimulate it with a single, you can't get it to contract, I guess, with a single stimulus. You need multiple high-frequency stimuli in order to get that to contract. You see it in a couple different places. One of the big places is this red racing stripe, right? So most of you would probably recognize this as a fish fillet. And this redder muscle right here is the tonic muscle. And fish have tonic muscle because many of them have to continuously move, right? And so it's this very aerobic type of muscle that allows them to have a different metabolic property than the surrounding muscles here that are lighter, that would allow it to move quickly to either, get food or to avoid from being food, right? So to avoid prey. The type of muscle you're probably most familiar with in general is called phasic muscle or the twitch muscle, okay? Now, the phasic muscle is a little bit different in its properties, and it's typically divided up into two major types, and then sort of one of the types has two subtypes. The first major type is called type 1, or sometimes called the SLO, or slow twitch, or slow oxidative. That's what the SLO stands for. These muscles are adapted to have a very aerobic means in terms of how they operate. They are good to do fatigue resistant exercises for like long distance running or cycling are examples of that. Cross country skiing, all examples of slow twitch muscle. So they have a lot of oxygen carrying capacity and a lot of mitochondria to help make lots of energy. The type 2 are called fast twitch. Now, fast twitch are divided up into two, and it used to be 2A and 2B, but now the newer terminology is 2A and 2X. So let's do the 2X first. The 2X are the fast, or they call fast glycolytic. We haven't talked about metabolism yet, but we can take glucose and very quickly, without the presence of oxygen, break it down into a little bit of energy, and that process is called glycolysis. So 2X fibers have the ability to make a lot of energy very quickly, but are very fatigable and are very powerful, but they can't go nearly as long. And so those are the 2X. And then in between those two is what they call the 2A. Those are intermediate fibers. So they're fast, oxidative glycolytic, or sometimes it's called fog muscle. These can be trained to be more like type 1 or more like type 2. So looking at kind of the stark differences, both of these runners are Olympic gold medalists. The one on the left, right, you look and it's almost no body fat, not much musculature, lots and lots of this slow twitch muscle, right, finishing a marathon a little over 2 hours, which is, 26, or a little over 26 miles. The picture on the right is a sprinter, so someone who needs a very powerful muscular movement for a very short amount of time, right? The Olympic gold medals in the 100 meters are won in less than 10 seconds. So they just need to generate a tremendous amount of power very quickly, while the Olympic marathoners, as an extreme, need to generate power for 2+ hours. So that's skeletal muscle. Now in between I put cardiac muscle because cardiac in some ways behaves like skeletal muscle. And you probably haven't broken it down this way, and there have some anatomical differences, but functionally it's not a bad way to break up the heart tissue between nodal tissue, atrial tissue, and ventricular tissue. So nodal tissue are the portions of the heart that can generate action potentials on their own. And it's just specialized cardiac muscles that can do that. You've probably studied these. This SA or sinoatrial node located in the upper right part of the right atrium, and the atrioventricular node, that's right at the junction of the atrium and the ventricle in the corner, basically portion of the right atrium, the bottom corner. And then also, not shown here, are nodes that are sort of randomly in the ventricles called ventricular nodes. And again, we'll hit those when we get to the heart and talking about its physiology and function a little bit more later. But nodal tissue is different than the atrial tissue. Atrial tissue make up the atria. They're not very thick. They're fairly thin in terms of their muscle fibers because they have to only squirt a small amount of blood from the atria to the ventricle. And one of the big things for most humans is most of the time that's downhill. So they don't have to generate much pressure, so they don't tend to be fairly big. And then the ventricular muscle, that's the one that has to generate the most pressure, especially the ones on the left side. So this is the left side over here. So what you're facing your right, that would be the left ventricle. This side would be the right ventricle. And it's hard to tell from this drawing, but the left ventricular mass is much bigger in terms of the muscle because it has to push blood all over the body, while the right just pushes blood to the lungs. So you have different tissues that have a number of different properties. One of the big examples is the difference between the atria and the ventricular muscle and its refractory period. Whenever a muscle or a nerve, they go into the category of what we call excitable tissue. Whenever a muscle or a nerve contracts, it has to reset, or fires, I guess better way, because nerves don't contract. But when a muscle or nerve fires, has an electrical event, it has to reset before we can have another electrical event. And so that resetting time is called the refractory period. And during the refractory period, it's either impossible to have another wave of excitement, or it's much harder. Okay? So the ventricular muscle has a much longer refractory period than other types of muscle. It's usually over 200 milliseconds. All right, so let's do a little math. It's not too hard. So if we had 200 milliseconds, that means my heart could only fire 5 times a minute, right? Because 5 times a second. 1,000 milliseconds, right, is 1 second. So 200 milliseconds means it can't fire for 1/5 of a second. If we have those 1/5 of a second times 60, you can't have a ventricular heart rate over 300 typically because of the refractory period. But the atria have a refractory period that's only about 100 milliseconds. So it's twice as fast, which means you can have an atrial heart rate over 300 and sometimes as high as 500 or 600 beats per minute. So why is that difference? Well, functionally, right, it's the left ventricle, as I kind of explained a minute ago, which is going to connect it, that pumps blood to the rest of the body, right? The right ventricle goes to the lungs, the left ventricle goes to most of the other body. If your ventricles quit pumping, you would die because you couldn't deliver sufficient blood and oxygen to the areas of need. If your atria don't fire, you're not going to be as well off functionally, but you'll still be alive because 2/3 of that blood from the atria to the ventricles are passive anyway, meaning we don't rely on the contraction of the atria to get it there. And so we don't have to worry so much about filling either. And the reason why we have this long refractory period for the ventricles is it allows the heart to fill. Because pumping the heart without blood in it doesn't do any good, right? Because the idea is to pump the blood to functionally serve the tissue around it. All right, so that's cardiac muscle. And then we have smooth muscle. And smooth muscle is called smooth muscle because you don't see any striations on the microscope, right? So going back to cardiac muscle, you can see striations. Skeletal muscle, you can see striations. Smooth muscle, I don't like this picture as well as the other one. No striations, all right? These darker areas are the nuclei, right? And not surprisingly, just like we had subsections of the other types of muscle, we have subsections of the smooth muscle. You have a single unit and a multi-unit. Single unit smooth muscle contracts as a single unit. And basically, if one of these fire, they all fire. So they're electrically coupled by what we call gap junctions. In your anatomy class, you might have used the word connections, is what some people use. And it allows that electricity to pass from one cell to the other, which allows a coordinated contraction. The second way that we can do this is what they call multi-unit. And multi-unit means it contracts as a multiple unit. Each muscle fiber in that smooth muscle system is independent. It's not electrically coupled. It needs to be recruited by a different nerve stimulation. And the more that we can contract, the greater contraction force we'll have. And a good place you see that would be the erector pili muscles, right? And so hopefully from anatomy, you remember erector pili muscle are the ones that cause goosebumps, right? So goosebumps have a fancy scientific name. It's called piloerection. Pili means bump, or that's where it comes from. And erection means to stand up, right? So piloerection is what we call everyday goosebumps. It's believed to be what we call a vestigial function. Vestigial means it's a remnant from some evolutionary past that we no longer need or incorporate into our more theoretically advanced versions. So as an example, humans have gill slits during development in utero, and we don't express those anymore, but they're considered vestigial. Some people consider like the appendix vestigial, but the appendix probably is pretty good evidence it's not. I mean, it's not a useless organ, but it's just part of our lymphatic system and immune system as well. So that's smooth muscle. So one of the things I think will be interesting is to compare the muscle types and kind of see how they're similar and different. So I mentioned the striations, right? Skeletal muscle is striated, cardiac muscle is striated, so they sort of look alike. Smooth muscle, no striations. That's why it's called smooth muscle. Control, okay? All muscle is reflexive, even cardiac muscle, okay? But skeletal muscle generally is voluntary control. Cardiac muscle, involuntary. You can't go, I'm just going to make my heart stop, right? It just goes. And smooth muscle is involuntary. So functionally, in this case, the cardiac is a lot like smooth. That's why I said it's nice to be in the middle. The shape of skeletal muscle tends to be long and cylindrical. The cardiac muscle is short and chubby. And I always, kind of laugh when, books describe smooth muscle as spindle shaped. And I don't think most of us, if I asked you the first day of class, draw a spindle, I don't know how many of you would draw something that sort of looks like a cigar. In terms of the nuclei, right? So nucleate, the skeletal muscle is multinucleate, meaning it has multiple nuclei per muscle fiber. Cardiac muscle is uninucleate, and smooth muscle is uninucleate. So they both have only one nuclei per fiber. The source for calcium to contract, because calcium causes all these muscles to contract, In skeletal muscle, it's intracellular calcium. It's all stored inside the cell, particularly in the sarcoplasmic reticulum, the lateral sacs, or some people call them terminal cisternae or terminal cisterns. But that's where we store the skeletal muscle. Cardiac muscle is both intracellular and extracellular, so it has calcium channels that help to bring calcium into the cell to aid contraction. And it also stores some of its calcium in sarcoplasmic reticulum. It's not nearly as extensive as it is in skeletal muscle, but both are operational. And then in smooth muscle doesn't store any of the calcium inside its cell, and it relies on extracellular calcium coming in through calcium channels to do that. So one of the things that affect the heart, as an example, and smooth muscle as well, would be caffeine. And what caffeine does is it does a couple things, but it opens calcium channels. And that allows more calcium to come into the heart, or more calcium to come into smooth muscle, which then increases the contraction strength and things like that. So some people, when they drink caffeine, they feel their heart do some weird things, and that's because of the extra calcium that's in the heart, which makes it more excitable. Probably the easiest way to explain it right now. I don't know if we'll have time to go into it when we get to that section, but maybe we'll mention it. In order for calcium to cause contraction, it has to bind to something. So in skeletal muscle, it binds to something called troponin, or some people say troponin. In cardiac muscle, it's also the same thing, troponin. But in smooth muscle, it's a substance called calmodulin. Fairly recently discovered, so it's actually got a pretty good name. Calmodulin just refers to the fact that it's modulated by calcium. So calcium binds to calmodulin, and that leads to smooth muscle contraction. We talked about refractory periods already. Skeletal muscle, we can talk about, I guess, in skeletal and smooth. Skeletal muscle has a very short refractory period, like literally 2, 3, 4 milliseconds, depending on whether it's a fast or slow twitch muscle. The tonic muscle is much longer, but those twitch muscles are pretty fast. Cardiac muscle, we said it depends on the type. In the atria, it's 100 milliseconds. In the ventricles, it's at least 200, right? So much longer than the skeletal muscle one. And smooth muscle is pretty short, not nearly as short as skeletal muscle, but much, 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 much shorter than cardiac muscle. Contraction controls the somatic nervous system. Cardiac and smooth muscle contractions controlled in multiple levels, hormonally as well as nervous-wise too. Contraction direction, skeletal muscle contracts very quickly, relaxes very quickly. Cardiac contracts fairly quickly, relaxes quick enough. Smooth muscle takes a long time to contract, a long time to relax. So that's my smooth muscle. So that's like a nice little comparison that you kind of should look at. Let's skip the sliding filament theory. I'm going to try to shorten this section up a little bit for you guys. And this is something you probably looked at in anatomy. And so we're not going to worry about it. Not that it's unimportant, but it's one of those things I feel a little more comfortable skipping because you probably learned it. So you wouldn't have to worry about this whole idea of excitation, contraction coupling, and what happens in order to contract a muscle. If we were actually going to be responsible for this, I would make you learn all these steps, all those steps. But I do want you to understand the last couple slides on muscle. So there are a couple types of muscle contractions, and so we're going to divide them up into two major types, and then the one major type is going to have two subtypes. So an isotonic contraction, that iso means same and tonic refers to tone. So what we have is it basically translates to a constant tension. In an isotonic contraction, the muscle develops force, that's what contraction is, but it also is changing length. Okay? So it's changing length, it's getting longer or shorter, and it's developing tension, it's contracting. The first type, and this is probably the type most people think about first when they think about muscle contractions, what's called the concentric. Con means towards, centric, as it sounds, center. So a concentric contraction is going to make the muscle move towards the center. The muscle is going to shorten. An eccentric contraction is when the muscle lengthens. So eccentric means, in this case, to get longer, right? Eccentric contractions are actually very helpful from a exercise point of view. Bodybuilders knew this for a long time, but it took science a while to finally catch up. So when you lengthen your muscle, you do a overload of it at a much greater rate typically than when you do with the concentric contractions. So eccentric contractions help to overload the muscle and cause better gains in both strength and muscle size, as an example. Some bodybuilders call that doing negatives. The second type, actually the third type if you think about it that way, is called an isometric contraction. Again, iso, same, metric refers to length, right? That's how we measure things. And so this means we have a constant length. So the muscle is developing tension, but it's not changing length. So it's contracting without a length change. And that's because the tension on the muscle exceeds the load. All right. Sorry. Let me rephrase that because it's backwards. The load on the muscle exceeds the tension you can develop. And so, go to the wall of your house, and hopefully it's sturdy enough. Don't break anything. And if you pushed as hard as you could on the wall's not going to move, right? Your muscles are going to stay in that contracted state, and that's called an isometric contraction. So, here's kind of a way to look at them as well. Here's somebody doing bicep curls. So starting from the bottom and going up, right? The biceps are shortening to bring the weight up. That's a concentric contraction. To bring them back down, right? To go from this up position to this down position. So to bring them back down, you have to lengthen your biceps muscle. That's called an eccentric contraction. And if you're just holding the weight level at a 90-degree angle to the floor, that would be an isometric contraction, right? You're developing tension, because if you didn't, the weight would fall down, right? Or at least your arm would fall down. So you're developing tension, but there's no movement, so the weight's staying at the same level. So that's a good kind of way to think about the three types of contractions there. The other thing I want to mention, because we're going to talk a lot about metabolism, is skeletal muscle needs ATP, right? I think we all know that ATP is the energy currency of the cell. It allows the cell to do cellular work. It's needed for a couple things. It's needed to caulk the myosin head. In order to get muscle contraction, we skip that whole section, but I hopefully remember we need to cross bridge. We need to take the myosin head, put on the actin molecule, and then have the myosin head ratchet, move a little bit, and that causes contraction. In order to get the myosin head off the actin and detach it, we have to put ATP back on it, which then recocks the head. The actual most energy needed in muscle contraction is to do active transport. It's to resequester the calcium that was released for the contraction in the first place. So we have to take that calcium and we have to move it against its gradient back to where it's stored in the lateral sacs of the sarcoplasmic reticulum. In addition, in order to have the muscle contract, it has to open sodium and potassium channels primarily. And in order to keep sodium and potassium where they belong, we have to pump those against their gradients as well. So we need ATP for a number of different things in order to fuel the cell. All right, so that's going to finish this lecture, and we'll pick up with part 2 and start with immunity next.